Hello, my name is Sasha. Today we will look at the role of non-coding RNAs in human disease. This is a big emerging field, and understanding their effects can be used as another tool in diagnosing or treating different diseases like different types of cancer, neurodegenerative, and cardiovascular diseases, as well as developmental, autoimmune, metabolic, psychiatric disorders, and more. Today, I'll discuss how non-coding RNAs affect Alzheimer's disease, more specifically, a long non-coding RNA called BC1 and how it affects spatial learning and memory in Alzheimer's disease. I'll tell you about a novel mechanism discovery that works by affecting amyloid precursor protein translation, as the paper title suggests. But first, let's take a step back and get everyone caught up on what is Alzheimer's disease, AD. Well, it's a neurodegenerative disease, and all that means is that the brain tissue degenerates and disappears, as illustrated on the picture, where a healthy brain is compared to the Alzheimer's patient's brain. AD has a certain hallmark where a bunch of amyloid beta protein plaques form. But what are these amyloid beta AB plaques, and what makes them happen? Amyloid beta is a protein that is normally a monomer, and it exists in AB42 and AB40 forms, where AB42 has two more residues at the C-terminus. One would get amyloid beta plaques from cleavage of APP, which stands for amyloid precursor protein. APP is cleaved by beta secretase and gamma secretase, resulting in amyloid beta protein monomers that can oligomerize into oligomers and then further into actual plaques. This is happening if there's too much APP floating around. Too much APP floating around, along with the missense mutations in presenilin 1 or 2 genes, which encode the catalytic subunit of gamma secretase, equal plaques, which is the most common cause for Alzheimer's. But why do the amyloid beta peptides oligomerize in the first place? Well, there are a few reasons. As I alluded earlier, the main reason is the impaired clearance of the amyloid beta. Another common reason is genetic mutation, just like the mutations in the presenilin genes, or the accumulation can also be a result of oxidative stress and even more general, like the lifestyle and the environment. So quite literally, how much and how hard a given person thinks throughout the day. Alright, so as I mentioned earlier today, we'll be looking at expression of BC1 impairs spatial learning and memory in Alzheimer's disease via APP translation paper. Precisely, we will look at what is the mechanism behind regulation of APP translation in the brain. Then, can this mechanism be used for identification or treatment of Alzheimer's disease? And lastly, what part of Alzheimer's disease are affected by this mechanism? In this study, the main hypothesis to answer is BC1 long non-coding RNA exacerbates Alzheimer's disease progression by inducing APP mRNA translation, leading to amyloid beta aggregation and subsequent spatial learning and memory impairments. So, how do we go about testing and measuring and actually getting the results? Here is the chosen methodology for the study. Mice are chosen as a small organism, amyloid beta peptides are measured, Maurice water maze used, reverse transcription, qPCR, Western blotting techniques are all used, BC1 binding and RNA fish assay, and finally, administration of RNA BC1. Now, let's go over some of these in more detail. To measure the amyloid beta peptides, the researchers took the samples from CA1 area of hippocampus. That's part of the brain in the center and it's kind of shaped like a shrimp or a seahorse. Then all components were separated by centrifugation. Then amyloid beta 40 and 42 along with amyloid precursor protein beta were measured using ELISA, which we all know and love from BCNB 301. And lastly, for brain secretions were stained using anti-amyloid beta antibody C42. Maurice water maze. What is it? It is a pool that is about 1 to 2 meters in diameter. It's filled with water that is colored to be opaque. It was invented in 1989 by Richard Morris. Somewhere in the pool, there's a hidden platform where an animal, usually a mouse, can stand on. This experiment is standard practice for testing spatial learning and memory in rodents. Why did we choose it? As I said, it's already a standard practice. It is good because it produces robust and reliable data, because unlike electrical stimuli or truth stimuli, this method uses natural stimuli, so wanting to leave and not wanting to drown. And as far as I'm aware, the death isn't one of the things that mice could build up tolerance to, unlike pain or treats. Since we will be talking about how BC1 affects APP translation, I think it's also important to talk about how and why we measure BC1 binding. It will give us mechanistic insights in the molecular interactions of it with other RNAs or proteins, which one we'll just have to see in the results section. By confirming BC1 binding partners, we get validation that BC1 can in fact be a therapeutic target. To actually measure it, we did fluorescence in situ hybridization targeting RNA, fish RNA. A quick rundown on how this assay performed in a paper like this. SDS precipitates BC1 and FMRP complex, then transfers to the nitrocellulose membrane, then it's blocked with blocking solution, and then we wash membranes 3 times. Lastly, BC1 probes are added like Cy3 tagged DUTP or deoxygenin tagged DUTP. Okay, so what is LNA BC1? It is a locked nucleic acid modified BC1, but in more general terms, LNAs are a family of nucleic acid analogues in which the ribose rings is chemically modified with a methylene bridge connecting the 2' oxygen and 4' carbon. This locked structure confers high thermal stability to the LNA DNA or LNA RNA duplexes and improves the affinity of LNA oligonucleotides for their complementary RNA targets. Examining figure 1, we see control versus Alzheimer's mice. In controls, levels of full-length APP and BACI1, a beta secretase, and beta-actin are relatively stable, indicating normal APP processing. In contrast, Alzheimer's mice show a significant increase in APP from 6 months onward, suggesting enhanced APP translation leading to excess amyloid beta. Yet, BACI1 and beta-actin levels remain unchanged, pinpointing the role of APP overproduction and amyloid beta plaque formation. Intriguingly, by qPCR analysis, figure 1H and I reveal a marked increase in BC1 RNA in Alzheimer's mice, underscoring its potential role in this process by showing a correlation with APP translation. Figure 2I shined light on regulatory mechanism of BC1 on APP mRNA translation. Our investigation centered on its interaction with FMRP. The segments of FMRP, particularly amino acids 61 through 79, this specific sequence can bind BC1, acting as a gatekeeper to regulate the interaction between BC1 and the full-length FMRP protein. After they conjugated the FMRP 61 through 79 segment into the HAV TAT1 peptide, known for its ability to transverse cellular membranes, this fusion peptide, introduced to Alzheimer's mice, precisely disrupts the pathological BC1 FMRP interaction. Observations from figure 2K and 2M confirm the efficacy of this approach, revealing a marked decrease in APP mRNA translation activity post-treatment. The inhibition of BC1 through LNA BC1 impacted spatial learning and memory in Alzheimer's mice. As depicted in figure 3A through C, this series of experiments used the Morris-Water maze to quantitatively measure the cognitive improvements plus inhibition. Initially, Alzheimer's mice inhibited impaired spatial memory, struggling to locate the hidden platform. However, following the administration of LNA BC1, a notable shift occurred. Figure 3A visually represents the trajectory patterns of mice in the maze, where post-treatment paths become more direct and efficient, mirroring those of healthy controls. These improvements suggest the restoration of spatial memory capabilities. Figure 3B reveals an increased duration spent in target quadrant, signifying not only the retrieval of spatial memory, but also its retention. Figure 3C used to address potential biases in the assessment of spatial memory. Swim velocities were analyzed and found to be consistent across all experimental groups, indicating observed improvements are cognitive in nature. In the Morris-Water maze, PNEBC positive mice engineered to overexpress BC1 showed notable lowering in spatial learning and memory compared to their PNEBC negative counterparts. Figures 4H and 4I eliminated these deficits through increased time and distance to locate a submerged platform. These mice exhibited a systematic pattern of extended search times and distances. This behavior is emblematic of Alzheimer's progression, with figure 4J's bar graph offering a visualization of exogenous BC1 positive mice spent significantly less time in the target quadrant, indicating a diminished recall of spatial cues. Again, figure 4K shows that despite these cognitive differences, swimming speeds were consistent across all groups. Figure 5a exemplifies this through quantified reduction in APP mRNA within treated neuronal cells, offering evidence of the intervention's targeted effect. Complementing this, Figure 5b portrays the increase in amyloid beta peptide production, associating with a decrease in plaque formation, which is a hallmark of Alzheimer's pathology. Figure 5c confirms findings by depicting the overall reduction in amyloid beta plaque within the brains of treated mice. Figure 5g shows a marked improvement in the cognitive function of treated mice. This improvement, quantified through enhanced performance in spatial learning and memory tasks, underscores the real-world implications of our molecular findings. So, in summary, let's quickly put everything together into one cohesive picture. BC1 RNA was found to be involved in the regulation of APP mRNA translation. This means BC1 can influence how much of the APP is produced inside neurons. FMRNP, a protein that binds RNA, typically regulates translation of various mRNAs, including APP mRNA. The association between FMRP and BC1 was shown to increase the translation of APP mRNA. When APP mRNA translation is upregulated, more APP protein is produced. This can lead to increased cleavage of APP by enzymes like BACE1, resulting in the production of more amyloid beta peptide. Going forward, understanding the new mechanisms in the study opens up new avenues for Alzheimer's prevention and treatment. The obvious strategy is inhibiting BC1 FMRP interaction or targeting BC1 directly. Delivery systems like viral vectors for siRNAs or peptide delivery mechanisms like ones for TAT FMRP offer promising methods for brain-targeted therapies. Exploring other non-coding RNAs beyond BC1, the roles of other non-coding RNAs in Alzheimer's provide fertile ground for research. MicroRNAs like MIR107 and MIR106B already linked to BACE regulation and amyloid beta production suggest broader RNA-based regulation in Alzheimer's. Similarly, long non-coding RNAs such as NEAT1 and MALAT1 involved in key cellular process like chromatin remodeling or mRNA transcription and translation. Alright, so that's about it. Hopefully you learned something new and enjoyed learning it.